Students, in this session we will learn about the structure of one more special type of chromosomes called as Lampbrush chromosomes. Last session we learned about the polytene chromosomes and its structure, and in this session we will learn about the structure of Lampbrush chromosomes. Now this is how a Lampbrush chromosome look like. You must be wondering why the name Lampbrush chromosome. If you look at the structure of this chromosome, you can make out it appears like the structure of a brush which we use to clean bottles and other stuff. So because of that it was called as Lampbrush chromosomes. We will discuss in detail these structures in the following slides. So in this session you will learn about the detailed structure of Lampbrush chromosomes, and I am sure you are going to appreciate the complexity of these Lampbrush chromosomes and its significance. So we learn the structure of these Lampbrush chromosomes under these following headings. First we will learn like where do you find these chromosomes, what could be their size and how they are formed, followed by its structure and functions. Now these Lampbrush chromosomes, they are found mainly in the oocytes of insects, sharks, amphibians, reptiles and birds, which basically produce large yolky eggs. At the same time they are also seen in invertebrates like sepia and echinaster, that is mollusks and echinoderms. So this was first discovered by Fleming in 1882 in axolotl larvae. These Lampbrush chromosomes, they vary in their length, and they generally extend up to 1000 microns in length, or it could be even more in certain species, and width wise they vary up to 20 microns. Structurally, if you see, it will consist of two homologous chromosomes which are in contact only at certain points, which we call it as chiasmata. Each chromosome of the pair is formed by two chromatids which lie parallel and form chromosomal axis or the main axis. Now this axis is differentiated into alternate regions of high density loops which are lightly colored and arise on both the sides of the chromosomal axis. The chromosomal axis, the chromomeres and this loop axis, they are all formed of DNA. The chromomeres are found in pairs, one chromomere on each chromatid. They are about 25 to 2 micrometer in diameter and are spaced about 2 microns from center to center along the chromatid axis. Now these probably represent the heterochromatic regions where axial filament remains tightly coiled. Now this will be clear to you when you look at these diagram wherein the Lampbrush chromosomes are shown. Here you should see it is made up of two chromatids, and then only at these points you find them being in touch, and these are all called as the chiasma, and in rest of the thing they remain separated. And in this region, when you enlarge and see, this is how the looping structure appears along the axis. Now this is the central axis around which the looping of this chromomere takes place, and because of these looping that it appears like a brush. Now here the size of the loops varies with an average of 9.5 micron in interchromomeric fibers, and these pairs of loops in these chromosomes give them this characteristic brush like appearance. Now it is said that infrequently these loops exhibit a thin axis which probably consists of DNA, one DNA double helix, and from which the fibers project, which are covered with a loop matrix consisting of RNA and protein. For example, if you just take this one loop and enlarge and see, this is how it looks like. It will have a the axis, this is the axis which is double stranded here, and then from which it extends the loop in the form of an extended chromatin loop. And here you can see the chromomeres which is formed when chromatin get highly condensed here. Okay, so this is how the loops will be formed. Now the electron microscopic studies of such Lampbrush chromosomes by Miller and Beatty in the oocyte of salamander, it showed the presence of dense granules on the loop axis of DNA, representing large molecules of enzyme RNA polymerase. Upon getting attached to DNA, these initiate RNA synthesis. Now from these RNA polymerase molecules arise fine fibrils of RNA which increase in length, and that's how the ultrastructure of a single loop has been described. Now each loop here is considered to be one long operon consisting of series of identical copies of the same cistrons, which are nothing but the structural genes, separated by a spacer DNA. Each gene locus probably produce a very long RNA molecule which interact with protein to form ribonucleoprotein. Now this is what we said so far. When you take a loop here, when you take this loop structure and enlarge and see, this is how this is the visible structure of a loop. We said that this will be the central axis, and then this is the loop axis around which you can make out this loop matrix, which are in turn made up of RNA. Okay. So this is the inferred structure, that this is how it could have arranged. Now the number of pairs of loops gradually increases in meiosis till it reaches maximum in diplotin stage. As meiosis proceeds further, number of loops gradually decrease and the loops ultimately disappear due to disintegration rather than reabsorption back into the chromomeres. According to one view, the loops were integral part of chromonemata, which are extended in the form of major coils. It is believed that these loops represent the modified chromosome structure at the loci of active genes. And it's also been observed that if the activity of these genes is stopped by an actinomycin D, which is nothing but an agent used to stop this synthesis of RNA on a DNA template, the loops of this Lampbrush chromosome will collapse, and suggesting that the loops are mainly consist of RNA. That's how we can say that this loop matrix is basically consisting of RNA. Now since the earlier, like when there was a confusion as to how these loops are formed and what actually triggers the formation of these loops, there were two main views that were put forward. One by Callan and Lloyd, wherein they said a chromomere is the master gene with solenoid supercoiling, which produces several identical copies of its own, thereby it results in loop formation. So these extend out as a lateral loop formed of linear strand of nucleotides representing the transcriptionally active stage, and these were called as slave gene copies because they make copies of their own. Now, according to the other view, which is called as spinning out and retraction hypothesis, a chromosome is fully transcribed from one end to the other by spinning out a transient loop. The new loop material spins out on one side of a chromomere at the thin end of the loop and returns to a condensed stage on the other side after completing the synthesis of RNA, and that's how it results in the formation of a loop on both the sides of the chromatid. So these are associated with rapid synthesis of yolk and protein in the maturing ovum, and these disappear by the end of first prophase when chromosomes become thick and more condensed, and because of which they thought that these Lampbrush chromosomes may play some significant role in formation of yolk material in the ovum. Now, what could be its functions? There are two main functions of this Lampbrush chromosomes. One is synthesis of RNA, because we know that RNA is present in each loop, and so it helps in production of this RNA. And then formation of yolky material, wherein there are some probabilities that these Lampbrush chromosomes help in the formation of certain amount of yolk material in the egg. So these two are the main functions of this Lampbrush chromosome. They play an important role in synthesis of RNA and formation of yolk material. So this is about the occurrence, the size, the structure and function of Lampbrush chromosome. So these are the books that are referred to pull in the information. You can go through these books again for any kind of clarification. And this will be asked as a 10 marker question, wherein you have to write the structure of this Lampbrush chromosomes, the picture that we saw, along with the explanation. So we'll just take a quick recap of few questions about what we discussed. In which typical stage are these Lampbrush chromosomes observed? Do you see them in the meiotic prophase, or do you see them in mitotic metaphase, or mitotic prophase, or mitotic anaphase? In which phase we find these Lampbrush chromosomes? They are mainly found in the meiotic prophase, so the answer is option A. Now, where do you find these Lampbrush chromosomes? They occur mainly in the oocytes, or in cancer cells, or in lymph glands, or in salivary glands. What's the right answer? It is option A, the oocytes. Loops in Lampbrush chromosomes represent site of. What do you find around the loops of the Lampbrush chromosomes? Is it a site of replication? Is it a site of transcription, or of cell division, or crossing over? What's the right answer? The right answer is it represents the site of transcription. Now the term Lampbrush chromosome was coined by Sutton, Hofmeister, Fleming, or Balbiani. Who found this termed this Lampbrush chromosome? Who coined this term? It's answer C, Fleming. Then functions of Lampbrush chromosomes are formation of yolk material, synthesis of RNA, both A and B, or none of the above. What is the main function of Lampbrush chromosomes? We said it helps in synthesis of RNA and it helps in the formation of yolk material. So the right answer is option C, both A and B. So that's all for this session. I hope this has helped you to understand the structure of Lampbrush chromosomes easily. So just go through the video again, and all the best.